All right. Hello, my microbiome colleagues out there. We are going to cover the first day that Microbiome Labs is truly a lab offering a gut test. Okay, so here we go. All right, so when you start with the test, what it will be like is like you're looking right into the microbiome of somebody. So here we have a healthy microbiome with lots of alpha and beta diversity. And then we have a patient who you can see is, if you take this yellow color as one type of organism, it's crowding out all of the other, the pink, the blues, and if they're representing different species. So you'll be able to see that this person is got a lot of symptoms. It's not representative of a healthy population. And then you'll have a summary sheet that will give you an index. You'll be able to see if this person is in the red or orange zone. It's because their alpha or their beta diversity scores are also in the red or orange zone, that their pathogen occurrence is in the red or orange, and their resistome, or the resistant plasmids that they have that confer antibiotic resistance, is in the red or orange zones. Then you'll have the job of designing a protocol to correct those indicators. We know that the metabolic endotoxemia is because these endotoxins in the form of lipopolysaccharide or candida exotoxins are breaching the epithelial barrier and causing rampant inflammation, that the diversity is low, and that the mucin 2 layer has been breached. So we have to go about designing a protocol to correct these problems. And we look at symptoms that would be related to lipopolysaccharide, and there are plenty. Chronic constipation, mood and appetite disorders, depression, and this, you see the description in these two different depression listings are different. Here you've got lipopolysaccharide migrating into the blood-brain barrier and causing inflammation, along with inhibition of dopamine receptors. And here you have lipopolysaccharide increasing the turnover of serotonin in the synapse of the central nervous system, reducing the concentration in those regions. Anxiety, chronic pain, Parkinson's, hypogonadism or low testosterone. So you do a complete history, you look at which autoimmune disease types they have, whatever indicators you're using to measure their symptoms. And then we go about the repair. So there's some tools that we're going to go over. This one here is the serum bovine albumin that is for the concentrated immunoglobulins of IgG 2000. And you see its job confers two different mechanisms to help neutralize toxins and pathogens. The first is immune exclusion. And as you can see here, by tightening up the tight junctions, you're not there, the pathogens and toxins are not able to pass through and enter the lumen and cause an inflammatory reaction. So that is immune exclusion. The second is steric exclusion. And here you have the immunoglobulins that are tagging, attaching to, or neutralizing pathogens and toxins. And then structure-wise, they're not able to fit through these junctions. If they do, they are bound, and they are not going to excite the immune system. This is a clinical trial that specifically shows the immunoglobulins and what they're able to achieve in the condition of IBS. Here you see a 39% reduction in loose stools, a 38% reduction in abdominal discomfort, 31% reduction in urgency, 29% reduction in bloating, 35% reduction in flagellates. Okay, so what other things can we do? We can pre-treat with the IgG 2000, arrest the lipopolysaccharides, exotoxins, and pathogens, and help shore up the tight junctions. But we also have some tools, as illustrated here in this clinical study of Megaspore. This was published in the World Journal of Gastrointestinal Pathophysiology, August of 2017, and it shows a 45 to 60% reduction in lipopolysaccharide. And this is a study here showing its performance in the condition IBS. Megaspore reduces the IBS severity score better than rifaximin with the nutraceutical and similar to the rifaximin on a low FODMAP diet. So very strong. And we know this from the SHIME studies. These are clinical trials that we do on a synthetic gastrointestinal tract. This is the most state-of-the-art way of testing probiotics. And that way we can see the exact mechanisms that are taking place enzymatically with microbial variables in this in vitro setting. If a person cannot tolerate all 5 strains of the bacillus spores in Megaspore, each one of these spores are very effective. And this clinical trial shows the 1 spore, which is Bacillus subtilis, in our product HU58, is very effective in reducing interleukin 6 by 45% and TNF alpha by 55%. We also know it drops ammonia amazingly, so much so that Cleveland Clinic uses it as their standard of care on their hepatic encephalopathy wing. Then we would take the 1 spore probiotic, HU58, and then graduate them to a 2 spore probiotic, which is in Restore Flora. This also has Saccharomyces boulardii. This is also very effective if you don't know how someone is going to handle Megaspore and don't have time to titrate them, but they are or have been put on an antibiotic or recovering from an antibiotic. This is very helpful. Okay, so we have some tools then for shoring up the tight junctions and helping with the diversity. And what's important in the diversity is to make sure you're supporting those keystone strains. We know there's great studies supporting Akkermansia, Mucophylla, Faecalibacterium prunitii, and a couple of different strains of bifidobacteria that correlate really well with good health, in that if they're not present, you have more inflammation, it's more difficult to lose weight, there's a whole host of immune problems. So we want to encourage those specific strains, and we have tested our prebiotic in that the synergistic study shows that Megaspore will increase the short chain fatty acids by 20%, but then by the addition of the prebiotic, you're increasing those very important keystone strains by 80%. So that's the way you're working on helping the alpha and the beta diversity in the microbiome. And then the final step of the total gut restoration is to add the mega mucosa, which is to rebuild the intestinal mucosa. And what that is doing is repairing this mucin 2 layer. So you're adding amino acids, you're adding more of the IgG 2000 and some citrus polyphenols to quench the oxidative stress. But those amino acids help the goblet cells produce mucin. And the studies show those amino acids will increase the mucin production by 95%, which is very significant. Okay, so the next thing we have to consider is the removal of toxins through the detoxification pathways. And I am proposing here to research the phase 2.5 detoxification. This is new information. It was published in the Townsend letter. It was produced by Dr. Kelly at the IAACN this past fall. Halterman, Dr. Kelly Halterman. And what she was saying was that once you have toxins that are being processed by the hepatic or the liver cells, you've got three primary protein transporters, and they will conjugate these toxins and toxic metabolites of drugs to bile salts with PC, and that's how you're transporting these toxins out of the body via the bile caniculi. But if you've got a lot of inflammation and dysbiosis, then you are unconjugating those toxins from the bile salts, and in severe cases, these transporters actually disengage from the bile caniculi, and all those toxins recycle. So we really need to support this system to make sure that it's effective and efficient. This diagram is just showing that most bile salts normally will travel through the intestinal tract and then be reabsorbed. And this is a very efficient system when it's working and not clogged up with bad diet and inflammation, drugs, and toxins. So we propose the artichoke in our formula. MegaGuard is really effective at helping that bile flow and making sure that phase 2.5 detoxification is happening. The other great components of this formula is the very rich flavonoid licorice that we have that inhibits the protein synthesis of H. pylori. It helps with the gastric mucosa pH balance, and then the ginger helps with gastric emptying and transport time. Okay, so some other things, unfortunately, that can impede these tight junctions is candida. And in this diagram, you see that candida grow hyphae. It's like roots that bury or dig or erode the tight junctions that also lead to leaky gut. So we have in our formula Mega Microbalance undeclinic acid, which is derived from castor oil beans, which is very antifungal, and it's very effective because it stops the hypha production. So the candida will pass through the gut. So then you avoid the damage that's going on. So this is very different than how the mechanism in how other antifungals work. And this is the formula. It also has some bee propolis. As you know, that's how beehives, protect their hives, because bee propolis is very antifungal as well. Like with all of our products, the formulas are very strong, and we recommend titrating everything, starting very slow. Okay, the other thing that will cause inflammation, we're finding more and more reports that all people are affected by wheat proteins that will cause inflammation. So our latest formula is Wheat Rescue. So we have these important enzymes that break down the wheat protein into non-immunogenic amino acids that will not cause inflammation. And then you see we also have some of our spores and Saccharomyces boulardii. So this is very effective at keeping the gut intact and working as a maintenance dose. Once a person is out on their own trying to find restaurants and shopping for themselves. So the hidden gluten will be processed properly. And then finally, I mentioned the vitamin K2 just because if we had a functioning microbiome that was fermenting the vitamin K2/7, it wouldn't be so much of a problem. But since we see so many gut issues and we're not getting proper absorption of our nutrients, and we're overusing vitamin D without vitamin K2, and we're mobilizing calcium, and then it's not being carboxylated. So it's staying in this uncarboxylated state and being deposited into soft tissue, and therefore it's not getting to the bones. So we're seeing hardening of the arteries, kidney stones, gallbladder stones, plaquing in the brain for Parkinson's, and osteoporosis. Now, there are about 14 different called GLA receptors that require vitamin K2 in order um, to be able to perform this reaction of um, putting the ionizing the calcitonin into the cell, into the bone. And here is our study. We took an approach of looking at cardiovascular effects of K2 and found that it increases cardiac output by 13% and increases blood flow dramatically. So this is very helpful for anybody, but certainly will give an edge to any type of competitive athletes. Here is the comparison between the two. The megaquinone is a formula designed for maximal absorption, but if you already have individuals on multiple vitamins and other products, it's recommended to use MMax so that you're not duplicating some of these minerals. And then finally, you would repeat the stool test and be able to see how well the products performed in a post-test study. So that concludes the presentation part. I'm going to go ahead and unmute so we can have some questions. Okay, how are you doing today?
I'm doing well, thank you. That was really interesting.
Oh, super.
I am a little bit behind in my emails, so I've seen the emails about the new stool testing, but I'm curious as to how it works, and I apologize if that's already in my email, but how do clients provide you with the stool?
Okay, let me pull up another slide that might be more informative. I can get this to exit. All right, there we go. See if I can find what I had this morning. Okay, well, I'm gonna try to talk you through it since you know how it is when you're trying to find something.
Right, especially when people are watching. Yeah.
Yes, but I will refer you to, I did a breakfast club meeting, and that's on the YouTube, the same one. I'll record, this is being recorded, and I'll send you this. So once you get into that YouTube, you can see more information there. So yesterday was the soft launch. Today, the kit is available. It's on the website. So when you see your other products, you would order it right along. It will be shipped separately, but if you order it with products, there'll be no shipping charges. Okay. So you get a packet of 4 different tests, at the same time. They're just trying to economize on the shipping by doing it that way. And there'll be a requisition in there that you can either pay for the stool test. It's $299, and then charge your client whatever you would like, or you can give the client the kit and then have them pay the wholesale price. It's at that point, it is up to you what you want to do with it. The test has a specific type of coring that's very different than other stool tests. Part of the problem is getting a representative sample, and just by scooping the outside of a stool or wiping methods that are out there is really not efficient, because you're only getting the colon area. So there is a tool, it's called a coring tool. It kind of looks like a mascara brush. So it maximizes the surface area and has you core 10 to 12 times the stool. So you're getting the inside and you're getting a better representative sample of the DNA. So that's a huge difference. The other difference is that other technologies are based on 16S sequencing, and that's what is being amplified in the PCR. So that'll take you down to a species and a genus, whereas the whole metagenomic sequencing is going to take you down to what would be representative of as a social security number. So the 16S in a phone book will take you to their last name, but the whole metagenomic will actually give you their social security number. So that's really the difference in degree of how much more specific this test is going to be. So the 16S just looks at bacteria. Whole genomic sequencing looks at bacteria, fungus, viruses, and not protozoa. There's another organism that is in the amoeba family. So I'll have to get back with you on that. So that, and then the final difference is the amount of database that they have. Most have, in the degree of thousands. This database is tapped into 160,000. So it's going to be the most comprehensive, and that's important so that you can really get a representative picture of what a healthy population is.
Mm-hmm.
And that's where a lot of them fall short. So there's been several different advances in the specificity, in the efficiency of sampling, and in the comprehensive database that we imagine this test is going to be the state of the art eventually. And then this is phase 1, that's really looking at the populations and their diversity. And then phase 2, which will be launched sometime in the summer, will go more in detail to functional type of testing, looking for amounts of short chain fatty acids, beta glucuronidases, TMAOs, so it'll be a lot more functional in that way. So this test launching now should compete and beat out all the existing stool tests, and then we'll do even another advancement in the summer.
Well, that's really exciting. Thank you for that information.
You're welcome. And it's difficult launching a new advanced test that's never been launched before. So we're really depending on our customers to give us feedback. Let us know how it's working in the field. If because we don't want to fall on our face just coming out of the gate. So
right.
Yeah, we really look for all of your feedback.
All right. Well, thank you.
Welcome. Any other questions you had on the protocols or putting the products together? Have you had some feedback from your clients?
Not yet, but I definitely will provide feedback as I receive it.
Super, super. Well, thank you so much for joining me. I'll be available to you anytime. If you think of something, just email, text, or give me a call.
Okay. Thank you so much.
You're welcome. Take care.
Bye.
Bye-bye. Thanks.